Hey, hi to those who have already joined. I can see the numbers are going up. So we're just going to wait a minute or so for most people to be here, and then we'll make a start. Okay. Hi, everyone. Good morning, good afternoon, or good evening. Welcome to this Networking Fridays MBON webinar. I'm Sophie Siave, and I'm joined by our speaker, Yasmina, who I will introduce in a short while. But first of all, as I'm here representing POGO, the Partnership for Observation of the Global Ocean, and OBON, the Ocean Biomolecular Observing Network, I've been asked to briefly present the OBON initiative just to set the stage before we have the presentation from our main speaker, Yasmina. So I will share my screen and I will go ahead and present the OBON initiative. So OBON, as I said, the Ocean Biomolecular Observing Network, is a program endorsed by the UN Ocean Decade of Ocean Science for Sustainable Development, and it's led by POGO. So POGO is an international organization I'm the CEO of, which is working on global ocean observations. OBON's vision, in a nutshell, is to accelerate informed decision-making to restore the health of our ocean using the universal signatures of life on Earth, biomolecules. Yasmina will explain a bit more about biomolecules and eDNA. Our mission is to create a hub for biomolecular measurement of marine life. You can use this link, and there's also links being posted in the chat to read more about OBON and particularly to find out about our strategy. But I just want to briefly summarize our strategy, which is centered around these 4 objectives. So to develop the networks and resources needed for biomolecular observations at the global scale, to innovate, particularly the technology, things like autonomous samplers, also looking at technologies for deep ocean observations of biomolecules, to enhance information use through particularly data management and data standardization, to eventually be able to feed these data into things like digital twins. And the ultimate sort of enveloping goal is to inform policy and management for a sustainable future. So being a UN Ocean Decade program, we have a number of UN Ocean Decade endorsed projects that sit underneath our umbrella, and we participate in the biannual calls for new project endorsements that are issued by the decade. So now we have 24 active projects within the OBON family and also 4 completed or emeritus projects. I know there's a lot of text here, so I won't dwell on this slide, but I just want to highlight where the projects sit in terms of the eDNA sampling pipeline, so the kind of process from sample acquisition through to processing and archival, data processing and curation, and finally data visualization and sharing. So we have projects that sit into these broad categories of observatories and time series. Some of them also do community science or citizen science. And some of them are, rather than being single observatories, they're large regional networks of observatories. So these sit at the sample acquisition and sample processing end of the pipeline. Then we have some projects that are focusing on improving and expanding regional reference libraries. And then there are some projects that are more at the downstream end of the pipeline that are looking at linking eDNA with fisheries or with MPA management. And we also have some projects that are supporting all the other projects with things like providing guidance on protocols for eDNA, systems for sharing protocols in a standardized way, and also for sharing data in a standardized way. So quite a range of different projects that we have. In terms of geography, we have quite a global scope and representation. There are, of course, quite a few gaps to fill, and particularly gaps in the open ocean, where most of our projects are sampling at coastal sites. In terms of our organization, so POGO is the lead organization and hosts the program office. We are governed by a scientific advisory committee and an executive committee. Both are chaired by Margaret Lynan from Scripps. We're in the process of establishing a user advisory committee to really help us with that sort of policy engagement part of our objectives. And we have 6 working groups that bring together our projects and work on common issues that are relevant across many of the different projects, such as technology development, capacity development, protocols and methodology, reference libraries, data synthesis, and policy engagement. And finally, I just want to highlight an exciting new project that we have funded by Minderoo Foundation. It was launched at the end of last year. Its vision is to achieve integrated autonomous global eDNA observations to address key scientific challenges to restore the health of our ocean. It has a strong focus on developing capacity in the Western Pacific region. And through this project, we're able to provide opportunities to our projects and working groups through things like microgrant funding calls, which are also very much focusing on supporting projects in the Western Pacific region. So again, all the information on that is on our website, and there's been quite a few opportunities for engagement from the global scientific community in this project. So that's it from me, just some social media and contact details here if you want to find out more about OBON, and of course feel free to get in touch with me directly. So now we will move on to the main part of this webinar. I'm just going to stop sharing my screen. And find my other window. Okay, so now we'll have the presentation from Yasmina Esmaeili. I hope I'm pronouncing that okay. So Yasmina is a molecular ecologist at the University of Texas at Austin's Marine Science Institute. She focuses on biodiversity assessment, invasive species detection, trophic ecology, and molecular resource development for long-term ecosystem monitoring and conservation efforts. And importantly, for this webinar and for regional collaboration, she's a co-founder of the Gulf eDNA Network, which is a collaborative initiative that brings together researchers, agencies, museums, and stakeholders across the Gulf of Mexico to advance biodiversity monitoring and molecular ecology research. So very much aligned with the goals of OBON. We're focusing on the Gulf of Mexico. So without further ado, we will hear from Yasmina, and I can see your presentation already. So please, the floor is yours.
All right, great, thank you so much. So you said my presentation is showing, Sophie, all good? Yep. All right. So good morning or afternoon, everyone, depending on where you are in the world. Thank you for joining me today. Special thanks to Joanna for setting this up and Sophie for moderating the session. As she mentioned, my name is Yasmina Esmaeili, and I'm a researcher associated with the University of Texas Marine Science Institute, and as well as a visiting researcher at Texas A&M in Galveston. Now, throughout my career, I have had the opportunity to explore a lot of marine biodiversity in some amazing places around the world. From the rocky reefs in Sydney, Australia, to the beautiful sandy beaches of Brazil, which is also where I first got involved with MBON during the MBON Pole-to-Pole workshop. Now, I've been in the US for a couple of years now and have studied ecosystems from the kelp forests in Maine to the coastal habitats of Texas, where I currently conduct most of my research. And in all of these places and different ecosystems, the focus of my research has always been to understand how patterns of diversity are driven by both environmental and human impacts. Now, through these research projects, I've explored many ways to assess biodiversity and how it differs or changes in time and space, ranging from extractive methods such as sediment cores and seine nets to different visual observation methods such as underwater visual surveys and baited remote underwater video stations. And in the slide, I'm showing you all of the nice pictures of the great days out in the field, but as many of you that have done biodiversity assessment might be aware, is that it often comes with some challenges. So we don't always have great visibility days, especially if you work in more coastal turbid habitats. So you can see a little comparison here of what Instagram would show that our research looks like, and oftentimes the reality on the right-hand side here. And we are also not always able to see all of the diversity for other reasons, such as huge kelp fronds getting in the way of some of our photoquadrates. And we know that all of these are filled with cryptic species that are hidden in between. And in addition to that, if we are going underwater, we're also often limited by things like time and oxygen if you're diving. And oftentimes we have to sample in conditions that are not always ideal. So depending on where you are sampling, it can be freezing cold, or for most of the places I have worked, very, very hot, or even outright dangerous, as you can see here in this video of sampling in surf zones of sandy beaches. In addition to that, unlike some of our scientific forefathers, we are not often offered year-long grants to sail the oceans and explore some of that biodiversity that is out there. And in most cases, we need answers and solutions as fast as possible, and doing so always staying within budget, of course. However, many of the problems we are facing today in relation to biodiversity loss, for example, are not limited to specific geographic regions. So how can we study this biodiversity at large spatial scales and over long periods of time when our time and our resources are often limited? Well, I believe the answer to that is on two fronts. On the one hand, with what you might have guessed, leveraging on some of the technologies such as the use of eDNA, as well as collaborations, so where we share data, expertise, and resources across institutions. But why eDNA? Well, if you think about it, we are basically surrounded by genetic material floating around us everywhere and at all times. And because all living organisms leave traces of DNA in the environment they frequent, with the advancement of technology, we are now able to determine who is roaming about, in this case, for example, our body of water that we are interested in. And so this gives us the opportunity to approach biodiversity assessments from a whole new angle. Now, I mentioned using eDNA or environmental DNA. So environmental DNA refers to all genetic material found in the environment, such as tissue, skin, scales, that we can use to gain information on individual species, which I won't focus on right now, but also on biodiversity by targeting specific regions of the DNA to learn more about target taxonomic groups such as fishes or plants, or to target groups of broad organisms such as eukaryotes or prokaryotes. And DNA has several advantages. First of all, it is a non-invasive method, so non-extractive really, which can be particularly of interest when dealing with pristine habitats or endangered species. It is also sensible to capture some of those low abundance or cryptic species that I mentioned earlier that are hiding in plain sight. And we also have, to a certain extent, fewer restrictions on where we can sample, because visibility, for example, won't play a part here, and we can access some of those more difficult to sample areas as well. And so overall, because of the overall ease that I will explain to you in a little bit to sample, the initial steps, they don't require a lot of technical knowledge, lending itself as a great potential tool for large-scale monitoring. So how does it work? Here are some pictures of my wonderful colleagues at work, but I will take you step by step through the process. So we have our organisms that are releasing DNA in the environment, and we can capture that DNA by collecting a sample of, for example, water or soil, extract the DNA from this sample using specific extraction kits. And then this DNA is amplified using PCR reactions. So basically, we make lots and lots of copies of the DNA. And then using a sequencing platform, we read in all of that information. And through further bioinformatical processes, we translate all of that information into a long list of individual sequences for every unique piece of DNA that we can then match to specific species. Now, this last step is particularly important because it relies on reference databases, which you can basically compare to a dictionary of known DNA sequences that are linked to known species. And so you can compare it often with barcodes in a supermarket where you scan an item, and based on this unique code, the computer links this barcode to a specific product. So conceptually, that's how you can think about how this last part of the process works. And as I mentioned before, eDNA has great potential for detection of biodiversity, but also targeting specific species of interest in the environment, ranging from rare species or endangered species or invasive species. And it has been used across several applications, anywhere ranging from the detection of toxic algal blooms to biomonitoring, including some of those difficult to access environments, as I mentioned. And now, because the ease of the sampling, it is, as Sophie mentioned earlier as well, really lends itself well for citizen science projects. Basically, anyone can scoop up some water and some soil. And this even further emphasizes the concept of how something that simple in these initial stages really lends it to scalability. And that's why, of course, there has been a growing interest all across the world, including the US and Europe, in terms of policy, as well as pushing for both standardization in sample collection, sample processing, and the creation of these reference libraries to keep improving our results. And with an increase in the interest, we also see an increase in the DNA sequencing market, where it's projected to become a nearly $75 billion industry. And with the growth in that market also comes a decrease for the consumers, of course, in the price per sample, which again makes it very interesting when we think about large-scale projects. Now, we know that not all that glitters is gold, and making something cheaper and faster does not always necessarily mean it will provide us with the best outcome. And as many of our other biodiversity assessment methods, eDNA is no exception to that and still has a lot of challenges ahead of us. So let's talk a little bit about those limitations. Well, first of all, with metabarcoding at this point, detection does not necessarily imply abundance values. We know that in the PCR process, this is a stochastic process, we can have some bias, so we can't necessarily link our amount of genetic reads to abundance values for organisms. And in addition to that, we also know that organisms shed their DNA at different rates. So whether you're a fish or a crab, you might be releasing DNA into the environment at a different rate. There's also a lot of research still being done looking at how stable this environmental DNA is in the environment, because we know that it will also be degrading. And a lot of that depends on environmental conditions at a certain point as well. Because we are dealing with genetic material, it is also very sensitive in terms of how it needs to be preserved. And of course, the collection of this DNA needs to follow streamlined or standardized protocols for it to be comparable, which is also similar to a lot of other methods, of course. And then the last point that I want to hit on is those reference database gaps. So we know that the genetic information for some of our taxonomic groups is still limited. So we still have a long way ahead of us to be able to fill those reference libraries with basically all of the diversity that we can find on our planet. And in addition to that, because a lot of these databases are public, there might be some sequences that are uploaded that are not necessarily always correct. So we do have some issues in that area as well. And also, we talked a little bit about genetic markers, and some of those markers are definitely still underrepresented as well. And this is important because, again, when you think about it, we are using this reference database as our dictionary to be able to link those sequences to specific organisms. And even though we see that markets or there is a huge growth over time with NCBI users or GenBank sequences that are being published, and this only goes to 2019, so probably we're already way up on this graph right here. More and more people are generating sequences of our fauna and our flora. But there's an additional layer that we have to consider here. And that is, first of all, that we are not there yet. If you've ever had to deal with a metabarcoding data set, you know that many times we have a lot of things that are unassigned or wrongly assigned, which has implications for how we interpret biodiversity, right? And this happens because we either don't have the reference for a certain organism available in our databases, or we don't have a local specimen in our database. And so the sequence will get matched to the closest thing it can find, which can end up being a species out of our spatial range. And a lot of studies have shown that if we use local or regional reference libraries that contain the organisms of interest within our region, the accuracy of those assessments increases significantly. And so again, we rely on local knowledge to produce these regional reference libraries. And again, that's where the taxonomists come back in. And for several regions in the world, we see that there is still limited genetic information available, right? Which means that with limited references, we lead to limited interpretation abilities of metabarcoding data. And that brings us back to our diagram that I showed earlier that emphasizes that in order to overcome some of these hurdles, collaboration becomes fundamental. And this is why, or one of the reasons why we created the Gulf eDNA network. So the Gulf eDNA network is a budding collaborative for eDNA research in the Gulf of Mexico. And before I go into a bit about what we do, let me introduce you to our team. So my fellow co-founders here, Dr. Sheila Kitchen and Dr. Jordan Casey, and our wonderful support team consisting of various talented undergrad and graduate students and technicians that really helped kickstart our network about a year ago. And so now that you've met us, what do we do? Well, our network is built on 3 pillars, which are to connect, collaborate, and cultivate eDNA research in the Gulf of Mexico. And I will start talking a little bit about our connect pillar, which is obviously us connecting with the different researchers in the region. And we did that by sending out a lot of messages initially, telling people about the establishment of our network, and reaching out to them, asking whether they are currently doing any type of eDNA research in the Gulf, or if they are interested in conducting eDNA research in the Gulf. And currently, we have about 125 members that range all across the US Gulf states and including Mexico. You can see that we have some additional countries and states listed there. That's because research in the Gulf is not always limited to the US or Mexico in this region. And we have researchers outside of the Gulf states also conducting research in the Gulf. And our partners go anywhere between 70 and 75 universities and different organizations that are already involved in our network. So if you are not part of our network yet, and if you would like to join our network, you can see the QR code that I've included here, which will lead you to our interest form, where you can basically fill out if you are doing any research currently in the Gulf, if you have any information, organisms that you're collecting, DNA sequences that you're barcoding, anything that you would be willing to participate in our network and share with different stakeholders around the region, you're very welcome to join us. And that brings us to our second pillar, which really focuses on that collaborate aspect. So forming partnerships between our different institutions and sharing knowledge. And one of the products that we have been working on so far links back to those regional reference libraries that I talked about. So one of the goals that we had was to identify underrepresented Gulf of Mexico species in genomic databases. Now, in order to be able to do that, to do this gap analysis that we will be talking about, first of all, we need to know what our Gulf of Mexico species are. And the way to do that is to generate a species list. So looking at some of the literature that is available, luckily a few years ago, we already had some colleagues that put in a huge effort to create a list of organisms all across the Gulf of Mexico. And so we built on that with additional information from databases such as worms, as I mentioned, literature, but also information from state agencies, as well as GBIF. So if you are not familiar on GBIF, you're actually able to draw a polygon across your region, and it will give you all of the records of occurrences of diversity in your region. So we use that as an additional layer to build on this species list. And so this species list basically forms the basis for our gap analysis for us to be able to highlight which species that we have and we do not have any sequences for in our databases. So what did we find? Well, an amazing diversity around the Gulf. Across all of the kingdoms, we found nearly 40,000 total species in the Gulf of Mexico list. As I said, pulling all this information from the databases. Now, because most of our expertise currently lies within the animal kingdom, we zoomed in on that a little bit, and we found that of all of those species, over 25,000 are animal species. And so what we did next was look at how these animal species are distributed across our habitats. And so you can see we are mainly interested in our marine realm. So this big chunk of blue that you see there are exclusively marine organisms. But we also have a lot of organisms that are from these different environments or occur in different habitats, anywhere from marine, freshwater, brackish, etc. And you will see that we also included, and this is where the biggest increase in our species list occurred in comparison with some of the previous studies that were done, is that we included a significant terrestrial part to this. Now, the importance of that is that we know, obviously, our terrestrial organisms are not living in the water, but their genetic traces do end up in our water bodies. And by including some of that terrestrial signature, we can improve some of those things that we might be finding that are unidentified match it back to those terrestrial organisms. And even though they won't be part of our diversity assessment, at least we can identify that they were there and then move forward from there. And so what we did next with this species list in hand was look at some of our genetic databases that are publicly available online. And I'm just going to highlight a little bit of some of the things that we found. So looking at GenBank specifically, for our top 6 most abundant phyla in the animal kingdom, we looked at how many of the CO1 barcodes are currently missing in GenBank per phyla. And as you can see in these little figurines here, we looked at anything from arthropods to flatworms. And most of it in the dark blue here, you will see for most of these groups, over 50% are still missing from reference libraries. Now, this is something really important to emphasize. When we are using this eDNA data and we're looking at our diversity, basically we're not capturing over 50% of that because we don't have the references available. And this is particularly so, we can see our chordates do a little bit better, but for some of our taxonomic groups, like our annelids and our flatworms that I mentioned, the situation is even worse in terms of what we have available. Now, when we zoom into that a little bit more, we made this little map where you can see all of the records that are available on bold in terms of CO1, filtered on the availability in the Gulf. So you can see this is our Gulf water body here in the light gray. And looking at some of our top phyla, which are arthropods, chordates, and mollusks, you can see that these are all the CO1 records that are available. But when we look at exclusively our aquatic taxa and the number of species that we have within each one of these groups, we still see it emphasizes even more that there is still a huge part of that missing in our reference databases. So a lot of the Gulf organisms that we are that we would like to have in our study are still missing from those reference libraries. And that brings us to the third point, and that is to expand our research with collective data, tools, and other resources. And so how can we together help solve this problem? Well, based on that gap analysis that we did, we created this most wanted list, and this is just a few highlights based on those GBIF records that we collected. We identified the most abundant Gulf species that actually at this point do not have references or entries available in our genetic databases. And so we created this list and sent it to all of our partners to see if we could obtain actual physical organisms to be able to extract the DNA from those organisms and then help basically increase the references that are available on these databases. And a huge shout out here to some of our partners in the region that have been fundamental in sending us both organisms, tissue samples, DNA materials, all of that, sharing sequences that they had available but weren't publicly available yet. So definitely want to thank all of them for what they have done so far. And this is also, again, a little shout out. If you do have organisms available from the region, please feel free to reach out to us, and I will share a little QR code with you later on how you can do that. But so all of these institutions have helped by sending us samples that we are able to process. And so moving to some of our next steps, obviously with these organisms in hand, we are now able to generate DNA barcodes for some of these underrepresented taxa in the region, with the ultimate goal of providing a regional reference library specifically for the Gulf that will serve all of the people that are doing eDNA barcoding in our region, and also create or leverage on some of those existing platforms to share some of this knowledge, right? Both in the form of our reference libraries, as well as our different partners that are collecting eDNA data. In addition to that collaboration, as I said, with these partners that we have, the 125, majority of that currently is academic institutions. And so we are looking into expanding to some industry, policy, as well as government organizations in our region to be able to have a broader reach. And also, especially when we are thinking about management tools for the future and how eDNA could be incorporated in biomonitoring, this is an essential part of that. We are also supporting research projects. So if you have a research project in the region and you would like to connect with us, or you're writing a proposal for eDNA sampling, we have provided several letters of support that can be fundamental for some of these projects that are ongoing. And we've also been putting a lot of effort into bioblitzes. So on small scales, we have been going around and trying to collect more organisms that we might not necessarily find in some of these collections. And I should say, especially for invertebrates, a lot of specimens have been preserved and that are available in our museum collections. They are often preserved in formalin, which there's a lot of research trying to look at how to extract DNA from those samples, but we are also trying to obtain a lot of organisms by doing these bioblitzes to be able to increase our reach. And then, of course, we are also applying for funding to maintain our network up and going. We still have a lot of work ahead of us. So we are exploring different ways in which we can keep our efforts going. And as I mentioned earlier, you can also collaborate with us. So whether you are doing work in the region or are just interested in seeing how our regional hubs function, you can join as a member, and this will give you access to our newsletters and email updates. So if you scan this QR code, again, that will take you to our interest form. And so feel free to reach out and share. We have, of course, our species lists and some of our reference database in place already. But this is, as you know, an ever-growing project, right? It is not, it's not done until we've reached all of those 40,000 species on the list. So if you have any species lists, organisms, sequences, or if you're interested in joining projects that are on this regional scale, again, please feel free to reach out to us. And you are also welcome to join our team. As we continue to grow, we definitely need more help. So if you'd like to be part of our leadership team or help create some of the materials that we have published, we are very interested in expanding our reach. And especially if you are a taxonomist, we definitely need more help with curation of our species lists. We're planning on forming specific working groups for some of our taxonomic, larger taxonomic groups that will help us only improve the quality of the products that we are producing. And then maybe another plugin of interest, we are currently considered a regional hub for the eDNA Collaborative, which is a global organization as well. And we will be present at the Global eDNA Conference in Seattle, Washington, where we will be hosting a session that's going to focus on eDNA research that is being done in the Gulf. And we will also be having a session with our partners that are going to be present at the conference. And we're also really interested in having other people outside of the region join this conversation so we can share experiences. So if you have collaborated in a regional effort, please feel free to come and talk to us while we are there. This is going to be the end of October, still a little bit of time. But today is actually the last day to submit abstracts for the conference. So if you haven't made the decision yet, definitely something that I would recommend. And with that, I would like to thank MBON again for having us today, for giving me the opportunity to share all of this with you. And I'm very happy to take any questions.
Great. Thank you very much, Yasmina. That was a really great overview of eDNA and what the issues are, and then also really nice to hear about your network, and especially the results of that initial study that you've done. I see a couple of questions in the Q&A. So one is from Brazil. Daniela is setting up, or her group is setting up an eDNA laboratory in southern Brazil. And she's asking, given the high sensitivity of eDNA methods, what do you consider the most important precautions to avoid contamination during sample collection, filtration, DNA extraction, and laboratory processing? And second part of that question is, in your experience, what are the most common mistakes made by new eDNA laboratories? So quite a big question.
Yes. And I understand the concern. I also attempted to set up a eDNA laboratory in South East Brazil. So I know that it comes with an additional layer of, oh, I see the question disappeared. So I wanted to make sure that I address all topics. Oh, I can find it here. Okay. So important precautions, definitely keeping everything as clean as possible. But luckily, that is not necessarily that hard to do. Bleach is your friend. If you start, if you are able to have a space that you are separating from all of other sample collections that are happening, I think this is something that is fundamental. So if you're able to dedicate a specific area of the lab or a specific closed environment that you can have isolated from anywhere that you are collecting actual physical organisms, I have found that that has often, from experiences from colleagues as well, the issue, if you are bringing in physical specimens into your lab and at the same time you're right there next to it extracting DNA, the risk of contamination is often pretty high. So I would say if you have the possibility of separating those spaces, even if it's, with some tape on the floor that says no crossing, that's how we used to do it when we don't necessarily have separate spaces, having that distinction between where you are doing your filtrations and extractions, I think is really important. And then the most common mistakes, I think it's not necessarily a mistake, I would say, but there are already a lot of resources available out there, right? In terms of protocols, sampling design, all of that. And I feel like people don't necessarily know that that information is available. So I wouldn't necessarily call it a mistake, but looking for that information that is already available, not necessarily trying to reinvent the wheel and do things differently. We sometimes have to adapt due to the conditions that we are in. But I think really taking a deep dive into how other people are collecting in this information, reaching out to other people for them to share the experiences, right? I think that is something fundamental. So I wouldn't call it a mistake, but definitely something that I would encourage everyone to do.
Great, thank you. So next question from Deepak is regarding which sequencing method was used for the work you did in the Gulf. So I guess your specific lab as opposed to the network in general.
Right. So it really depends on what we're looking at, right? For our eDNA data from my lab specifically, we do next generation sequencing. So we use Illumina platforms to basically look at general markers such as 18S, CO1, 12S. And then for the reference libraries, the organisms that we are receiving and that we are extracting DNA from, we are doing whole mitogenome sequencing. So that means we have information of the entire mitochondrial genome available to us, basically, for those organisms that we are sequencing. I hope that answers the question.
Great, thank you. I'm sure he can email you maybe if he wants more.
Definitely.
Details or links to references. Next question, considering necessary amplification beyond monitoring the microbiome and other biases mentioned, how much sampling and sequencing would make eDNA monitoring representative, at least qualitatively, of the overall diversity worldwide?
Yeah, that's a good question. I think, of course, we always know the more the better, right? And I think the key really is in the fact that we cannot by ourselves do everything, right? So if we, again, can find ways to standardize how DNA is collected, and we can do that across several regions, and in these small hubs basically collaborates, then I think that can really increase, what we can see on overall diversity across a global scale. And there are many initiatives that are trying to do that. And so I think I'm not sure if that answers the question, but sequencing or sampling in general, if we are collecting water samples for a specific region, usually it's recommended to have anywhere between 3 to 4 liters of water that is filtered. And so if we can have at least that initial bit of information from all of these different places, that would really help us get a bigger picture view.
Great, thank you. That you tackled that difficult question very well.
Yeah.
The next question from Frank regarding both OBON and the Gulf eDNA network, and how people, particularly people who want to develop similar networks, can go about doing that, and also linking with other networks. So yeah. Sorry, maybe just I can say from my thought listening to your presentation was that what you're doing at the scale of the Gulf of Mexico would be a really great thing to replicate for other people to replicate in other regions, to develop those regional reference libraries. And OBON can perhaps help with that kind of translation or, replication of existing great initiatives, and fostering those collaborations and that transfer of knowledge. So from OBON, I would say just email us in the OBON Secretariat, and a great way to get involved in OBON is by applying for endorsement as a UN Decade project under OBON. And Yasmina, from the Gulf eDNA network perspective?
Yeah, I think this is, again, highlights the importance of what we call having champions, right? People that are really pushing things forward on a smaller scale. If you bring all of those smaller networks together, it is so much easier to do things at a larger scale, right? And we found that with the Gulf network, we are talking about a very small chunk of the world. But if you have some key players, some people in place that are really passionate about making things happen, joining forces, then everything is so much easier to move this forward. And so I really, for starting up a similar network, I think it comes down to having people. People play a crucial part in this. And so if you want to start a similar network, we are very happy to share some of our experiences of doing it at a local scale. What were some of the challenges? How did we try to overcome this? Very happy to share that information with other networks, and also connect to those other networks, right? Because I think what we are doing on a smaller scale can then be replicated by other small networks. And that, again, gives us the opportunity to expand beyond our regional hubs.
Great, thank you. Another question is, one of the important issues is in which way eDNA enabled data conserve biodiversity monitoring, one would start by comparing presence-absence data from both the conventional taxonomic approach and from the eDNA approach without the blasting of sequences to species, and compare the emerging patterns, inferring process, and perhaps consequences from changes. Can you comment on this?
Yeah, of course. So we have a lot of studies, especially because eDNA is still a growing field. A lot of the literature that is being published at this point is comparing, actual physical sampling or any type of survey methods that have been used in the past and comparing that data to what we get with eDNA, right? And oftentimes the result of that is that it is highly complementary. So I'm definitely advocating for the use of eDNA, but the use of eDNA is only useful if you already have some information on your system already, right? Because a lot of it relies on really knowing what your system already looks like in order to then moving forward be able to use it as a useful tool. So I definitely advocate for the combination of eDNA with some of our traditional approaches. And especially, of course, as was mentioned, at this point when we are doing meta barcoding, this doesn't necessarily give us, we basically will be working with presence absence data, especially if you are combining multiple markers, for example. And I think that it is important to have, I think we're getting there. At some point, we will be able to get more than presence absence data. But since we are not there yet, and if that is something that really is important in terms of, you need actual abundance values, then definitely maintaining that balance between different methods is crucial. Mm-hmm.
Great, thank you. Quite a few questions still. I hope we can get through them all. So Christopher says, I think you said around half of the 40,000 barcode IDs in the Gulf have no reference barcode in libraries. So of the half with reference barcodes, do you have an estimate of what proportion of the references are voucher specimen based versus those that might be erroneous? He said that they're doing a great deal of taxonomic work to establish voucher-based libraries, and it would be helpful to understand where the biggest gaps exist.
Yeah, of course. So this is a very important point that you touched on, Christopher. Thank you for that. And we are actually looking at that as well. I didn't have the time to paste that in here, but luckily, oftentimes when those voucher specimens are published, they have a specific voucher code with them. So we are running our gap analysis, also looking at all of those barcodes that we do have available, how much of them are actually linked to voucher specimens. And so if you want, please feel free to reach out to us. We'll be more than happy to share some of that information with you. And help out to really look at those patterns to see where some of that is lying. But I would say if we compare, just as I mentioned earlier, chordates versus inverts in general, I would say the biggest gap is probably from those invertebrates, because we are still limited to a certain extent by the preservation methods that are often used for soft-bodied organisms.
Okay, thank you. And a question about eDNA in Africa. How can eDNA be exploited in Africa, considering the low turnout and the financial burden in low-income countries, Africa being a place where eDNA has yet to be done?
Yeah, and I think that is a very valid question. Even though we talked a bit about how the cost of monitoring using eDNA can somewhat be overcome, I know that it is really difficult in certain places of the world to have access even to things like extraction kits and things like that. So I think leveraging on international collaborations, having, of course, we can't solve all of the problems in the world through our perspective, but if some of higher income countries could invest in helping some of these places in terms of providing the resources, forming the partnerships that would really help uplift other regions in the world where, as you mentioned, eDNA is still to be done. I know how difficult it is to do it in places where you don't have access to all of this. So again, leveraging on collaborations is, again, definitely going to be the answer to that, I think.
Yes, thank you. If I can just add to that also from OBON, we, as I mentioned earlier, with this SAGE project funded by Minderoo, we're focusing at the moment on the Western Pacific region. But we have had several requests and people in our network who have asked us, what about Africa? So we're planning and trying to source funding to do something hopefully similar, or run some training workshops and try to support capacity development in Africa as well. So to anyone who's interested in that, again, if you want to get in touch with OBON or follow what we're doing, hopefully there will be some opportunities coming up. Another question from someone who says she's not an expert in the eDNA, but is interested in exploring it. If eDNA is used as a tool to analyze the presence or absence of species, how do you mitigate the possibility of detecting DNA that has been washed in from species that are not actually present in the area? And related to that, how long is eDNA typically detectable in water? And how does the persistence of DNA affect your confidence that a species is actually present locally rather than represented by DNA transported from elsewhere? That's a question that comes up a lot.
Yes, that is a question that comes up a lot and is very valid. People are often skeptical when it comes down to, we know we are dealing, most of our water bodies are flowing, right, from one place to the other. And so there has actually been a lot of research looking into, if you have these habitats close to each other, how do you know with the water flow that is coming in and out that the things you are looking at are actually present in that specific location and not coming from somewhere else? Well, first of all, you will have an influx of things that are coming from somewhere else. But when you think about it, if organisms that are living in that specific habitat and are in those surroundings most of the time, they will be the ones that have the highest basically shedding of materials in that specific environment. And so studies have shown that even though you might have some organisms that pop up that are being transported from somewhere else, or the DNA is transported from somewhere else, your signature will be highest for the organisms that are actually there and frequenting the whole time in that water body. Now, there also have been studies looking at some of combining what we know about our eDNA and then looking at some of the hydrodynamics of the water. So usually they've shown that depending on environmental conditions, eDNA can persist anywhere up to 24 hours in the water column for us to get a valid signal of it. However, if we can combine some of the information that we get from eDNA with models of how our water bodies are moving, that is definitely going to increase some of that confidence that we have about what we are seeing, if it is actually from the location that we are looking at.
Great, thank you very much. I think that's the end of our questions, and we're also 2 minutes from the end of the session. So I think if it's okay with the organizers, we will close the webinar here. And thank you again very much, Yasmina, for the great presentation and for tackling all those difficult questions. And thank you to MBON for inviting both of us and for putting on this this great webinar series.
Yep, thank you so much. And again, I left my contact information here. You can also reach out to us via the emails that are provided, or just go to our website. And we're very happy to share any information or experiences that we've had for our region that might be applicable to anywhere in the world.
Thank you. And Joanna has posted the link to the recording for anyone. I know a few people were asking about access for colleagues who weren't able to attend. So once again, thanks very much. Thank you to everyone for joining and for all the interaction. Goodbye and have a good weekend.
Thank you.
Thank you very much. It was a great session. We still have
people
here with us. Thank you, Sophie, for the moderation, and Yasmina for the presentation and answering all the questions. It was very interesting.